Okay, well, I hope you aren't that afraid of chemistry, because that's a little bit about what we're going to talk to today. But my name is Steven. I'm a senior here studying biochemistry, and the title of my presentation is Modulating Selective Protein Chemistry. I'm grateful to have done this in the Schwab lab over the past couple of semesters and past 2 summers. So to begin, I want to start with a more common example that might be familiar to you of selective chemistry that you might encounter on a daily basis, and that has to do with bread. Bread, under the right conditions, think the right temperature, the right amount of heat, the right equipment, can transform into toast. Using this idea, proteins, less of these, but more proteins like this, in our bodies, also with the right conditions, the right temperature, the right equipment in our bodies can undergo selective chemistry as well. This, I hope, is familiar because it's on the poster, but this is an example of ubiquitin, which is an important protein within our bodies that is involved in degradation of proteins. I will go into more about why these areas are highlighted, but I wanted to first bring your attention to this idea of a protein as opposed to the food that we're eating. So with that in mind, like I said, because proteins can be modified, can be selectively changed in the way that bread to toast can, they can also be affected by carbohydrates. Thinking again about bread like this. Now we're going to go into a little bit of chemistry, so I hope that's okay for everyone. But now I want to think about carbs more as sugars like, okay, that's not working. Sugars like this. Our body is able to use sugars, carbohydrates, in order to produce energy, and therefore allow me to speak today, allow you to kind of pay attention to what I'm saying, hopefully. And in the process of producing energy, of producing ATP, sugars transform into a bunch of different metabolites. They take on a bunch of different transformations in other chemistries. Metabolites such as this one on the bottom featured in red can interact selectively with different proteins. After interaction, they can undergo a series of different changes and lead to different structural changes to certain proteins. This scheme overall reflects the chemical process that I've been studying for the past 2 summers. Can you hear? Should I use the microphone? Okay. Just making sure. Thank you. So this reflects glycation overall, which is a process wherein proteins, such as this figure on the left, interact with sugars and these sugar metabolites, and then undergo a series of changes in order to produce some type of impairment within a cell. So basically, add this sugar metabolite, and you kind of mess up the protein possibly. The interesting thing about this too is that this is occurring spontaneously within this cell, so it could be happening at all times. With that in mind, glycation itself is implicated in a bunch of different changes in different proteins like these ones featured here, and they're implicated in a bunch of different disease states for different diseases, whether it's Alzheimer's, such as this protein in the middle, lens crystalline on the right, which is associated with your eye and your ability to see, and it is also used as a clinical marker for diabetes. These areas here that are highlighted reflect areas of the protein that would get selectively modified. So one thing that the Schwab lab has been concerned with is trying to understand how exactly this process is occurring, where is it occurring on different proteins in the cell, and how can we figure that out? How can we develop methods to answer this question? So we've been able to understand in lab that this change is occurring selectively. So take this protein again, ubiquitin. These highlighted areas reflect sites that are able to be glycated via this reaction, or able to undergo this chemistry. However, this certain purple one that is reflected there, that certain part of the protein, R42, is able to demonstrate a higher level of glycation than other areas on the protein that are highlighted. So basically what that means is that area will be affected the most relative to the other ones. We want to understand why exactly is that happening. It remains extremely difficult to do this because the biology for this, as it's implicated with these different diseases that I mentioned previously, aren't fully understood. It's more like, oh, we think this is probably contributing, but there's no this leads to that causality. So it makes it a little difficult to understand. There are numerous bunch of different factors, some of which I'll mention in the research that I conducted. There are a bunch of different sites on a protein that can be reacted with. There are a bunch of different agents, such as those ones reflected on the right, that can undergo this chemical reaction with different proteins. And there are also a bunch of different outcomes that can be seen. We do understand, however, that the molecular environment, if you will, when you look at a protein like this, the environment, I'm just going to point because the laser pointer's not working too well, but the environment that's created around these sites is unique. And it could be that environment that's there that is leading to a specific outcome. So how do we understand that when we have a bunch of different sites on one protein, and then we also have a bunch of different proteins in general? Beyond that, as well as I mentioned, there are also a ton of different agents that can lead to glycation. So these two major ideas, having a ton of agents such as these, this is not exhaustive, these are just the 4 ones that I was able to study in lab a little bit, as well as the complexity of places that can get glycated, make it very difficult to understand. So with that foundation, that was the basis of the research that I did. And I attempted to answer the question, how can we determine where glycation is happening and why it's happening in 2 major ways. One was to investigate these glycating agents. That was done by using a peptide, which is similar to a protein. I don't want to go into a full discussion of protein structure, but I want you to take away that this is similar enough to a protein and allows us to draw conclusions about how glycation is happening. Using this peptide, I looked and reacted with a bunch of these different agents with the peptide in order to try to understand what products and adducts are forming. I did this with all of these 4 here. The top one, methylglyoxal, has been the most studied and was used mostly as a control during these experiments and was able to reflect a diversity of products across the different aldehydes, across these different reactive glycating agents. So these are just an example of different adducts that we saw. So different products that were possible after this chemical reaction. I know I went through that fast, so I just want to highlight the point here that you can see that a diversity of products is evident here, but is not so much evident here, here, or here. And these are also all the product of multiple replications. You also will notice possibly too that because there are different time points here, over time, the type of adduct that you're seeing is changing. So that's perhaps also another layer of complexity in trying to understand how this reaction is actually occurring because things are changing over time as well. Beyond screening these reactive agents and trying to understand what products are seen represented by these numbers here, I'm sorry I didn't explain that previously, I also tried to take our hypothesis about glycation and apply it to ubiquitin. So what I was trying to do is take our understanding of what we think might increase glycation or decrease glycation and reflect it in a protein. Be able to change a part of this protein and say, if I make this change, I think that I'm going to increase glycation, do experiments, and then say, yes, look, I validated this hypothesis. Or the inverse as well. If I include something that I think will decrease glycation, I think I said increase, then be able to reflect that I also decrease that. So one way in which I was attempting to do that was by taking ubiquitin, like I said, looking at another part of the protein of another part of ubiquitin and see if there's a way that I could change it to affect what chemistry is going to happen at that area right there. Do this by mutation, introducing a mutation to that protein, such as here, changing this green structure into this other green structure, and then screening those reactions. So this slide here is to reflect kind of the progress that I made with that. These are different proteins that I was able to create. These just represent changes to the protein structure that I was able to affect with mutations. After doing this, the next step would be to run through a standard protocol in lab to then again try to see what products are forming. With that, thank you for paying attention. I want to especially thank obviously Laidlaw, Diana, and Dawn for allowing me to talk to them a lot, and for Professor Rebecca Scheck for giving me the opportunity to do this research in the first place. I learned a ton, and I'm super grateful to have had the chance. So thank you for listening, and I open the floor for any questions.
Questions? Anyone?
What do you mean by that?
Basically look at the structure on the top of this protein over here and how that structure is different than this structure on the left. So the structure on the right would be what I would consider a product for this reaction. And that's just like one possibility of many.
How did you decide which residues you wanted to mutate when you did it?
So to jump into the chemistry for that, and a lot of this came from like the planning that had already been done in lab and not necessarily just my planning alone. But the idea was to look at, so I'll go back to here. So look at some of the residues, maybe here's better. The purple highlighted residue is the reactive residue that would get attacked via this reaction. So look in the space around the protein, or around that reactive site, whatever's within, I believe, like 5 Armstrongs was what was used as like a measure, and see how we could change those residues under the idea that the ones that are closest to it in 3-dimensional space are probably part of the mechanism by which this reaction is happening. So then using trends that were observed in lab that I didn't really go over here because it's much more chemical than I wanted to broach, change them into other amino acids that we think would increase or decrease glycation. Those were discovered by, or those were determined by a series of peptide reactions.
Any other questions? If not, I've got one. So particularly when you're looking at ubiquitin, is it? It is glycation, part of the normal cell process? Is it, something that it would be seen, to happen normally in the body?
Yeah, so it's something that occurs normally and occurs over time. I guess one thing that I also neglected to mention, unfortunately, I should be using this, unfortunately, was that it also is implicated in the aging process. So because there are always sugars in our body and they're always turning into different things and there are always proteins, during the normal life cycle of a protein, sugars are going to interact and changes are going to happen. The thing is that this has been shown to be, in the literature, demonstrated with certain disease states, and it's not clear exactly how that's implicated.
Okay, so by understanding the path or modulating glycation, you're able to better understand the aging of cell.
Yeah. So in theory, by possibly discovering some type of mechanism or some type of study or method to like investigate this, we might be able to get insight into exactly what products are forming and then connect that to specifically aging, or connect that to like, oh, because this formed, it has to be degraded or something like that, if that makes sense.
Yeah, thank you very much.
Thank you.